Okay. Hello everyone. I am Beas Mukherjee, a research scholar from Dr. Shobhoday's lab in Department of Bioscience and Biotechnology at IIT Kharagpur. So this is going to be our third day of the week 1 question answer session of the NPTEL course Fundamentals of Protein Chemistry. So I think two other scholars before me have been discussing question and answers with you guys. So you might already be familiar already. Right, so have you attended the previous sessions or is this your first session? You can answer in the chat box. So in this session, we'll go through some of the topics that were discussed in week 1 and then we will simultaneously solve questions. Okay, these are all questions ranging from very simple things to a little complex. So you can stop me wherever you're facing any problems. So we'll wait for 5 more minutes for everyone else to join and then we'll begin. Okay, until then I'll give you a very brief overview. So in this first week, you have been taught about amino acids, right? And then peptide bond and very basics of this spectroscopy. So we'll touch upon all of these and I have tried to make the slides simple and informative because you will be able to download all of these slides and practice from here. Okay, plus in case you're missing any of the sessions, there's nothing to worry because all of these recordings are there on YouTube. If you want to refer to the previous year's recordings also, those are also uploaded on YouTube. Okay, so I hope you will find this informative and useful. And if you have any doubts, please reach out to me. All right, you can type in the chat box or you can unmute yourself and you can ask me any questions. If I'm unable to answer, I will go back and look into it and then get back to you the next day. Okay. You guys can talk. You guys can talk to me. There's no like you can always chat in the chat box. I'll read your messages and you can even unmute yourself and say if you have anything to say. All right. Yeah.
Hello. Ma'am, yesterday the Google Meet link had expired and later on we were not able to join again. So will there be the same issue today also?
Sorry, can you repeat yourself, please?
Yes. Am I audible?
Yeah, you are.
So yesterday the Google Meet link had expired after the 1 hour,
Okay.
and later on I was not able to join the same meeting. So I was thinking that I've lost some of the lecture yesterday.
Yeah.
So will that same be happening today with the lecture meet getting expired again?
Right. I'm not sure if those will be the same questions because the previous day's session was by another person, right? But you can always find that person's videos on YouTube. So the
Yeah,
YouTube
yeah, sure.
link will be shared to you. So in case you
No, no. I'm just asking that is there a time limit for today's meeting also? Because
yeah,
if this meeting gets ended by 1 hour, then we won't be able to join by the same Google Meet link
no,
that
no,
has been
there will
provided
be
to
no
us.
such time limit because this is Google Meet, right? So the Google is no time limit as such.
Okay, ma'am. Sure, it works.
Today's session, it is going to be around 1.5 hours. So normal time span for us is around 2 hours, but I know how bored you people will be. So we'll quit after 1.5 hopefully, and that's it.
Sure, ma'am. Thank you.
Right. So let's start with it because a lot of you have already joined. So other people will be joining in too, but then we can start with the work, right? So, okay. So this is for, once again, I'm saying this for everybody. Do not worry if you miss out or if there is any kind of interruption in between the meet, because all of this is going to be uploaded on YouTube, so you can refer to it once again. If you have any questions, please type in the chat box, or you can ask me by unmuting yourself. If there's any question that I'm unable to answer, I'll get back to you the next week. All right. Okay. So let us begin. Tell me if you're not able to see the slides or you're not able to hear me, because here the connection sometimes gets a little disturbing. Okay. All right. So let's begin. So for this today's session, we will start with the basics of things that you have learned in the week 1, right? So that's amino acids and peptide bonds and little bit of stereochemistry of amino acids, a little bit of spectroscopy, and a lot of questions, both numerical questions and normal MCQ questions. All right, so hopefully this is going to be useful to you. Right, so let's begin. Okay, now I'll be discussing a little bit of theory and then you'll be solving a little bit of questions so that it doesn't get very boring. Okay, so firstly, let's talk about amino acids. Now, amino acids are organic moieties that join together to form proteins. They are joined by peptide bonds. Now, amino acids are amphoteric, meaning that they have both a positively charged group and a negatively charged group. So here the positively charged group is definitely the amino group and the negatively charged group is the carboxyl group, right? That is why they behave as an acid as well as alkali depending upon the pH of the media. Now, amino acids, they can be proteogenic amino acids, meaning that they form proteins by joining together, right? There can be non-standards or non-proteogenic amino acids, meaning that they are citrulline, ornithine, these all amino acids, they are involved in the urea cycle. So these are also amino acids, but then they do not form proteins. All right, they are involved in other metabolic pathways. Now, amino acids are very important for different functions in our body. We will have a look at that very soon. Okay. Now, amino acids can be essential and non-essential. So, essential amino acids are these which can be. So, these essential amino acids are required by our body because they cannot be synthesized.
Excuse me, madam.
Yeah, sure.
Can you please make the slide full screen?
Right, right. Is it not full screen for you? I have actually presented. Okay, 1 second. You tell me if it is all right. Okay.
Okay.
Yeah. Is it okay now? It's full screen, right?
No, it is not changing. It is similar to previous one. Not changing.
No, 1 second, 1 second. Any problem? You tell me. I can't hear you properly. Now you tell me if you can see it as full screen or not. All right. Is it full screen for you?
It is not full screen, but it's not problematic also.
Okay, it is not full screen, right?
It is going on.
1 second, 1 second. Then I'll make it full screen mode because sometimes because of the slideshow, you are not able to see because it doesn't change as fast as I'm changing it for you guys. Right, so sometimes that's a problem. So now hopefully it will
Okay.
be better. Okay, right. So now, what are these essential and non-essential amino acids and what are semi-essential amino acids? Right, so the amino acids that can be synthesized by our body and they are obtained from diet. These are called the non-essential, sorry. So the ones that are synthesized by our body, these are called non-essential amino acids, and the ones that cannot be synthesized by our body and they are required from our diet, these are called essential amino acids. Now, what are semi-essential? Some amino acids are produced by our body but in very minute quantities, and therefore we need supplementation from outside. These are semi-essential, right? So this is very, very basics, but then sometimes you get MCQs from here. That is why it's better to remember. Now, amino acids are very important. Why? Because they help in all of these different functions, such as the hormones and brain neurochemicals. They are made up of amino acids. Amino acid is required for building muscles, boosting the immune system, and normal digestive processes, etc. Now, here is a list of some of the amino acids and their role, okay, in different functions of the body. Now, this was taken from a particular article. The link to that article and the entire citation is provided in the slide, so you can go back to it and you can read up. All right, but this list is important because sometimes in some exams people can ask what is the role of these individual amino acids, right? And this can come in the form of diseases. Okay, so read about this. Now, there is this quick question for you. Do you think now everybody is very concerned about all of these protein supplements and amino acid supplements, right? So, according to you, do you think amino acid supplements are required by our body, or is it just a marketing gimmick? So, what do you think? Actually, the answers and all are visible. So, I hope we could make it slideshow, but then you'll not be able to follow up if I make it slideshow. Anyway, so the thing is that from our normal diet, we get proper amount of nutrition. Okay, so the basic needs of the body can be fulfilled from the diet, the normal regular diet. But sometimes there can be deficiencies due to different reasons. Okay, for that maybe amino acid supplements can be taken, but normally for a normal healthy human being it is not required as such, because we get all of these things from our diet. Okay, now let's go. So all of this is very important because headaches, weakness, irritability, and then skin problems and mood disorders, then anemia, all of these things can happen if there is a deficiency in any amino acid, right? Now, amino acids and their properties. Now, this is very important. So, amino acids have been grouped into different categories based on their chemical nature. Okay, there can be non-polar. Now, there can be amino acids with non-polar side chain. For example, alanine. And there can be amino acids with polar side chain. Right. So, these are, excuse me. So, polar uncharged side chains, they have non-ionizable side chain. The entire list is given here, and this visual map should be sufficient for you to understand the different categories into which amino acids are divided. Okay, now amino acids with charged side chain, positively charged such as lysine, and negatively charged such as aspartic acid. Now, this is very important because the charges on an amino acid on in a particular pH gives charges to proteins, and because of these charges in the amino acids in proteins, the proteins become functionally active. Whether a protein is going to bind to DNA, whether it is going to bind to another protein or not, a lot of it is actually determined by the charge of its amino acids. Okay, and that is why it is very important to understand this. Okay, now we'll move on. Okay, so now another very difficult thing for students is to remember the structure of amino acids. But this is simple if you practice. The only thing is you have to understand what is happening, and you just have to practice again and again so that you get an idea of how the different chemical groups come together to make different amino acids. Alright. So this entire chart is given to you. Go through this. And it's very simple. For example, glycine is the amino acid. It has two hydrogens, right? So for its side chain, there is only one hydrogen. Alanine, instead of that hydrogen, you just put in a CH3 group. What happens in valine? There is a bifurcation like this. So this is how you can try to remember all of this. Right? It's simple. Just practice more. and that's it, because this is necessary, because for pH titration and all, you will have to remember the structures of these amino acids. Okay, now there's this quick question for you. Try to answer this. You can write the answer in the chat box. Okay, so which of the following sets contain all polar neutral amino acid residues? Polar neutral. Now think of the side chain, and you also have to remember the single letter code of the amino acids plus the triple letter code. Okay, because in questions either single letter code or triple letter code can be mentioned. Okay, so the first option is glycine, aspartic acid, asparagine, and glutamic acid, which is polar neutral amino acid. You can answer in the chat box. Okay, I'll keep the chat box open for you. Yes. Okay, the second option is FYW, STC, and D is IVLM. For the second question, you try answering the first one, and if you're done answering, we can go to the second question also. Okay, the following sulfur containing amino acid. This should be simpler. Bolo. Think about it and answer. This is very simple. We saw it in the last slide. Cysteine and methionine. Okay. A. This is for question number which one? 1 or 2? 1A. Okay. Polar neutral amino acids. Acha. And for the second one is 2A. Okay. Let's see. Right. Okay. So for the second one, the answer is methionine and cysteine. Right. For selenocysteine, this is actually a variation of cysteine. This is found in bacteria, some bacteria specifically. Here what happens, instead of sulfur, there is selenium. Okay. And for this one, so number 1A, this is your what? Glutamic acid. Glutamic acid is having a COOH group in its side chain. So it is not polar neutral. Okay. So see, this is glutamic acid. It should be here. Right. So here this is glutamine. So instead of this NH2, there will be this COOH. Right. Okay. Here you can see it's given properly. Right. So just refer to this and you'll understand. Right. So now we go to another interesting thing, which is stereochemistry of amino acids. Now, in nature, normally amino acids are L amino acids. Okay. But in some bacteria, there is D amino acid. Now, what is this D and L? So, this is based on the location of the chemical groups, that amino acids are D amino acid and L amino acid. This is different from dextrorotatory and levorotatory. So dextro and levo means dextrorotatory is rotating light, plane polarized light towards the right, right? That is dextrorotatory. And levorotatory is rotating plane polarized right towards the left. That is denoted by small l and small d. But this L and D is actually the Fisher projection formula. Okay, and see, capital L and capital D means describing the absolute configuration of a molecule that predates R and S but is still used for some biological molecules such as sugar and amino acids. L and D have no relation to optical rotation of a molecule. That is what I told you just now. Right, so when amino group stays on the left of the left hand side of the amino acid, it looks like an L, and this is called L amino acids. So in nature, in our body also, all amino acids are L amino acid. But in some bacteria, in the bacterial cell wall, this peptidoglycan layer is there. In the peptidoglycan, D amino acids are also there. Okay, so one example is shown here for you. See, this is what it looks like in the peptidoglycan layer in bacteria. So this chain, pentapeptide chain is there, and this pentapeptide chain is where this particular D amino acids are there. Okay. Now, what is dextro and levo is explained here in better details. So see, light is there. And what is happening in normal light? In normal light, the electric and magnetic field vectors are oscillating in different planes, and then you pass it through a polarizer. So polarizer is like a small window through which only the light whose electric and electric field vectors are rotating in a particular, oscillating in a particular plane are allowed to pass. And then this is called plane polarized light. And then when you subject this to the sample of amino acid, it is actually rotating it, right? So that is dextro and levorotatory, right? Now we'll move on. Okay. So now this is a question for you. So which of the amino acids has the maximum number of chiral center? So chiral center means that, let's say this carbon is there and it is having 4 bonds with which different groups are attached. So if different groups are attached to all the 4 bonds of carbon, then it is called a chiral center. It's a chiral molecule. Only then it will show optical activity. That means it will rotate light. If, for example, glycine, there are 2 hydrogen, 1 hydrogen here, 1 hydrogen here. So then it is not chiral anymore. It will not rotate light at all. Okay. So here they are asking which amino acid has maximum number of chiral centers. Now for this you have to remember the structures of the amino acids. Now which is the amino acid which is having 4 different groups attached to its carbons and maximum number of those chiral centers. You can try, otherwise I'll show you the answer. Isoleucine, serine. Now in serine there is only one OH group. So it is a very small amino acid. Right. In tryptophan there is a huge ring. This is an aromatic amino acid. So as you get used to the structures of the amino acids, all of these questions will become very simple for you. Right. But now that we are just starting, it will feel a little complicated, but then it will get easier. Okay. So let's go to the answer. Right, so see 1 and 4. So isoleucine and threonine contain the highest number of chiral centers, that is 2. So here and here. This is your C alpha and this is 1 chiral center and this is 1 chiral center, right? Here there is also 1 chiral center and 1 chiral center. So that is the maximum number of chiral centers are there for this particular amino acids. Now we go to another concept, which is hydropathy index. So hydropathy index means that whether a particular amino acid is water loving or not. That is its hydropathy index. So what does this mean? You know about these proteins, right? When proteins fold, many amino acids are there towards the core of the protein. These are hydrophobic amino acid, meaning that they cannot interact with water, and that is why they are pushed into the core of the protein. This is called the hydrophobic core. And the amino acids on the surface, they are actually constantly interacting with water. These are hydrophilic amino acids, right? So what is hydropathy index? This is actually a kind of plot that tells you whether the amino acids in a polypeptide, they are more hydrophilic or more hydrophobic. If they are more hydrophobic, then the values are positive, as you can see in the plot A, and if they are more hydrophilic, then the values are more negative, right? So this is used to predict the behavior of amino acids in a protein, particularly how they will interact with water and how they will be distributed in the protein 3-dimensional structure. So the list of all of these different amino acids are given, and which all amino acids are hydrophobic? Mainly the ones that are having your non-polar side chain. They are most hydrophobic. Okay, right. And the ones that are having charged side chains or polar side chain, these are hydrophilic, because these groups, they can be charged and they can interact with water. Now I'll ask you a tiny question. Let's say there is this protein. Okay, it's a membrane protein, and the N terminal, that is the beginning of the protein, starts here, and then this is how it goes through the membrane. This is the cell membrane, and a part of it enters and then again passes through the cell membrane, again it goes out and again passes through the cell membrane. This is how it goes. Now 1, 2, 3, 4, 5, 6, these are all regions. What is topology? Okay, where did you find topology? Right. So topology has different meanings. Okay. Topology, I think it is there in this slide. I will be discussing it. Hopefully, we'll see. Okay. So membrane topology, it's there in this slide, membrane topology. Okay. So basically topology, it is like what is the composition of this, let's say this particular protein, right? So that is topology. You can look this up, but right now I cannot exactly remember exactly what to tell you, but it comes in different like, what do I say, different aspects it comes. Okay. So anyway, we'll see this. So see how it is distributed across the membrane, that is being shown here, right? So these 1, 2, 3, 4, 5 and 6, these are actually regions that are passing through the cell membranes. These are also the portions of the same protein. Okay, they are passing through the cell membrane. Now inside the membrane, we know that it is made up of lipids, and the lipids are having, the head of the lipid is hydrophilic and the tail of the lipids are hydrophobic, right? You have studied the fluid mosaic model, everyone, in school. So this is how the lipid membrane looks like. So all the regions inside the membrane, this is actually hydrophobic in nature, because inside the membrane it is hydrophobic, but outside the membrane it is hydrophilic. That is why when you look at the plot in figure A, you see that 1, 2, 3, 4, 5 and 6, these are all having positive values. They are all hydrophobic, but then the region that is actually uh, before this, within the part that is there within the membrane, so that is the N terminal and C terminal region. These are relatively hydrophilic, right? This is how you understand the hydropathy plot. Okay, now there is a question for you. Okay, you try to answer this question on hydropathy index. Okay, so identify the amino acid with the lowest value for hydropathy index. So lowest value means that it should be negative, right? So if it is negative, that means it is what, hydrophobic or hydrophilic? We saw in the plot. Try to remember the plot.
Hydrophilic.
Hydrophilic, correct, right? So then which one do you think should be? See, there are many that can be hydrophilic. There are actually, all of these amino acids are hydrophilic. So now to see which is the most hydrophilic, we will have to go to this table once again. And here you can see, and I think this does not have all the amino acids, but the answer for this question is actually asparagine. And all the values of the amino acids are given here, so you can refer to this and you can answer this question. Okay, right. So now we go to another interesting thing, which is isoelectric point of a amino acid. Now these are questions that are repeatedly asked in every exam. Okay, they will give you a tripeptide, they will give you a dipeptide. Dipeptide, if they give, it's relatively easy, but they will mostly give you tripeptide, or they will even give you a longer peptide, and they will ask you to calculate the pi of this particular peptide. Okay, so this is something that is a very simple concept. Once you understand it, it's very simple, but if you don't understand, it just messes with your head. So let's try to understand this, right? So another thing is, try to imagine it this way, that you take an amino acid, okay, and you place it in a pH of, let's say, 1, and then you gradually keep on adding a base so that the pH increases. Now what happens is, the charge on the amino acids in this peptide also keep on changing. Why is that? Because there is something known as pKa of an amino acid, right? So at physiological pH, most amino acids exist as Witter ions. What is Witter ions? Witter ion means that it is in a particular chemical state in which the net charge on this amino acid is 0. Net charge is 0 means that there can be positive groups, there can be negative groups, but the number of positive groups is equal to the number of negative groups.
वाह, लाके तो संभाली करो।
Something?
ତମେ ଯଦି ମତେ ନ କହିଛ ପୁରା ବିଲକୁଲ ପୁରା ବସିଥିବି ନ କହିବ ତମେ ଏବେ ମଧୁଆ କାଲି ଦେଖିବ ମତେ।
I think she is not aware that she is unmuted.
କିନ୍ତୁ ଗୋଟେ ଶିଖିବ କିଛି ନାହିଁ।
Right. So the chat box is once again open. If you have anything to ask, please ask. Right. Okay. So we were talking about isoelectric points of amino acids. Right. So what happens is at physiological pH, they exist as zwitterions. But before that, before the physiological pH, they are mostly positively charged. And after the physiological pH, they are mostly negatively charged. But why is that so? So, see, what happens is the pH at which the amino acid has equal number of positive and negative charges is called the isoelectric point or PI of the amino acid, right? Now at pH values above and below the PI, the amino acid bears positive and negative charges respectively. Okay, so let us look at this particular amino acid as an example. So when it is very strongly acidic, then what happens? This COO group, it is not ionized yet. So it is there as COOH. COOH has zero charge, correct? But then this NH3 plus, NH2 group, this is present as NH3 plus. And that is why it is actually positively charged. So how can you remember this? Let's imagine it this way, that when the pH is very high, that means the H plus ion concentration is, sorry, when the pH is very low, right, then the H plus ion concentration is very high because pH is negative log of H plus. So when the pH is very low, H plus ion concentration is very high. So that is why this NH2 group has accepted an H plus and it has become plus charge, right? But COOH group, this is uncharged. Then the pKa of COOH group is around 2 point something. So when you increase the pH to this 2 point something, or let's say 3, then what happens? This COOH group leaves one H plus ion and it becomes COO minus. Now what happens? See, at intermediate pH, there is this COO minus and NH2 plus, NH3 plus. Then what happens? It is existing as a neutral zwitter ion. Then as you increase pH further, this NH3 plus releases the H ion and it becomes NH2. Right. So this is what happens when there is only the main chain. Only the main chain is concerned. So here we did not consider the side chain. See, R group is given as R. There is no actual chemical like thing for R group here yet. Right. So the zwitter ion is a salt formed as a result of acid-base reactions of the basic amino group and the acidic carboxylic group. And so what happens is deprotonation of COOH, that is loss of H plus, and protonation of NH2 to give NH3 as pH decreases. So these are the two things that are happening when pH increases and pH decreases respectively. Okay. Now we will look at something called the Henderson-Hasselbalch equation. So this also you might have already studied in school. So Henderson-Hasselbalch equation tells pH is equal to log of pKa plus log of the concentration of the A plus, which is the your conjugate base, and the acid, which is the HA, right? So based on this equation, you can, so if the pKa is known and pH is known, then you can find the ratio of the amount of H plus that has been broken, that has been separated from the amino acids. So lower pKa value means stronger acid, and pKa is the pH at the midpoint of the flat buffering zones in the titration point, that is pH versus volume of titrate. So graph is given where you see that initially at lower pH the positive charge dominates, then the neutral charge arises where you get the PI, and then the negative charge dominates. All right. Now we will look at this with an example. So this is how you calculate the pKa and PI of individual amino acids. Okay, so let's say for a particular amino acid, aspartic acid, this is having a side chain also. For normal di, like let's say there is an amino acid with just the main chains. Main chain, 2 pK values are given, then Pi is equal to pKa1, pKa2 divided by 2. So just summation divided by 2. It's easy, but they will not ask this in the exam. In the exam, they will always ask you Pi for a triprotic amino acid, which is having 3 pK values. There is a side chain involved, right? So you have to remember the structure. So aspartic acid is having this normal COOH group and then NH3 plus group connected to the C alpha, and there is this another COOH group which is there in the side chain. So what you do is you will have to first determine, so to calculate the Pi of the neutral or diprotic amino acid, the average of pKa1 and pKa2 values are taken. However, to find the Pi of the triprotic, the average of the pKa values where the 2 pKa values, because of which you get the zwitterion, those 2 pKa values are only taken. The other pKa value is ignored. Yeah, sure. You have any question?
Ma'am, the PK values are different, so
Yes.
we will be provided with the PK values, or we will
Yeah,
have
you
to
will not.
remember them also.
No, it's too much to remember. You will normally be provided with the pK values, but for some amino acids whose pK values are very general, for example, for cysteine, the pK value is around 8. And then let's say for arginine, and then for these amino acids, since you will be practicing many times, you will kind of remember. Okay, so those might not be given to you, but normally the pK values are given for the amino acids.
Oh, okay, ma'am.
Okay, so now, yeah, so for the triprotic acid, 2 pKa values that are closest to the point where 0 pH comes, that, sorry, 0 charge comes for that amino acid, those two you'll have to take. So how do we find that out? It's very simple. See, for example, I'll not take alanine, we'll take this one as an example. Okay, so see what is happening initially when the pH is around 4, then this one gets ionized, and so this is actually COO minus, right? At 2, at pH 2, this one gets ionized, and this is around this is also COO minus. At pH 10, this one gets deionized, so this becomes NH2 uncharged. So that means between which two values is your um, 0 charge coming? You tell me. Between which two value? pKa of the, like, between which two pH? pH 10, pH 4 or pH 2? This is simple.
Repeat it again, ma'am.
See, for this particular amino acid, let's say aspartic acid, okay, at pH 2, this pKa of the side chain, this comes, that is why COOH loses one H plus, it becomes COO minus. So at pH 2, there is, the charge is, this is -1, and this +1 charge is there because of NH3 plus. Then at pKa 4, pH 4, the alpha COOH group loses H plus, and your charge is now 2 minus and 1 plus, so that is -1. So initially the charge was what? It was 0, now it is becoming +1, and then as you increase the pH, at pH 10, it is going to become -2, because this NH3 is losing this H plus sign, it is becoming NH2. So before pH 2, the charge was +1. After pH 2, the charge is -1. Then the charge becomes, one second. Acha, give me 2 seconds. You think about it. I'll come back. So did you think about it? So you will have to take the two pKa values which are closest to the pH at which it becomes, the overall charge becomes 0. So which one is it going to be?
pH2.
pH 2 is 1, correct. But two values are supposed to be taken. So at pH 2, the charge is actually 0, right? And one more is there, which is actually pH. Two pH values are to be taken, two pKa values are to be taken, between which the charge becomes 0. So one pKa value is 1.88 and the other pKa value is what? So see here the pKa values, actually pH values go this way. +1 initially. At pH 1.88 it becomes minus, no sorry, it becomes I think 0. Then it goes -1 and then it becomes -2, right? So +1 0 -1 -2, that is how the different pKa values go. So basically the two pKa values would be 1.88 and 3.65 because your 0 is there coming just in between these two values. That is why they are taking these two values, and see we are getting 2.77 as the PI. So as you practice, it will become simpler. Similarly, for the basic amino acid, the trick is that the side chain pKa value is, if it is acidic, it will be less than the pKa value of the main chain carboxylic group. And if it is basic, the side chain pKa value will be more than that of the main chain NH2 group. So then two values between that means the main chain NH2 group and side chain NH2 group. You're supposed to take these two and you're supposed to get the PI. That is how it is done. That is the trick. Okay. But the basic logic is this, and the trick I have told you. Okay. So now these are the different amino acids and their pKa values at a glance. Right. Right. These are again other amino acids and their pKa values. Okay. For example, histidine. This is very biologically significant. Okay, so some of these are very significant. For example, arginine, this has the highest pKa value in its side chain, which is 12.48. So these are some very basic ones that you have to remember. Bas, Baki, it's given here. You can go through this. Okay, now try to answer this question. So an amino acid with neutral side chain. Neutral side chain, the pK1 value is 3.4, pK2 value is 9.4. So at which value of pH will the amino acid migrate towards the anode? Now anode is positively charged or negatively charged? First you tell me this. How will you remember? Anion goes to anode, cation goes to cathode. Positively charged. Okay, correct? Because anion is negatively charged and therefore anode would be negative, anion, yeah, anode would definitely be positive, correct, right? So then it is saying that it will migrate towards the anode. That means it will be negatively charged, right? So at which pK value? All these different pK values are given. So first thing that you have to do is you have to find its PI. PI calculation is simple. You've understood by now. Neutral side chain means just two values you're supposed to take. You're supposed to find the average. Okay, done. Now, if this is the PI, less than PI, the amino acid is going to be positively charged. More than PI, it is going to be negatively charged. Then you find out based on this different pH that are given, at which pH the amino acid is going to be negatively charged. Okay. So 3.4 plus 9.4, what does that give you divided by 2?
Ma'am, is the answer 7.4?
Answer is 7.4. Okay, we'll check. For for the next question, try the next question now. So the two amino acids have R groups with negative net charge at pH 7. Okay, and so they are at pH 7, they are having negative net charge. Then think about the trick. What should be the pKa values of these amino acids? And from there only you can decide whether these are both are negatively charged or both are positively charged, or what is the characteristic, right? So we look at the answer quickly. See, so both of these are having net charge at pH 7, right? So that means if we go back to our previous slide, try to recall this example. Both of them had pKa values less than 7. That is why at pH 7 we had negative charge, correct? So then both of these should be acidic amino acids. Now we go back to our question once again. So yeah, this is the right answer, aspartic acid and glutamic acid. Okay, now for this one, yes, you're correct. So the right answer is A, which is 7.4. So their pi is actually 6.4, and so above the pi, the amino acids would be negatively charged. Okay, we'll move on to the next slide. Okay, so we have time. So now this is the kind of questions that you can expect in your exams. Be it
I'm gonna explain that second question. Can I explain that second one?
Yeah,
Yeah.
sure. So see, for the second question, they are asking you which two amino acids have R groups with negative net charge at pH 7. So negative net charge at pH 7. So pKa of the R groups of aspartic acid and glutamic acid is around 3.65 and 4.25 respectively. Okay, therefore at pH 7, so if the pH is less than, if the pKa is, right, so basically the pKa of the R group is this, so that means your pi would be much less than 7, right? Pi would be much less than 7, and less than pi, the charge of the amino acid is negative. More than pi, it is what? Sorry, less than pi, it is positive, and more than pi, it is negative. So at pK 7, then these two amino acids would have negative charge, right? Because the pi is very low. Got it? So they have overall negative
Yes,
charge at pH 7. Okay, now we'll move on to the next question. So this is the kind of question that you can expect in the exams. This is the titration curve of a triprotic acid. Okay, so they are asking you, this is for arginine. Now the pKa values are all given. pKa values are given, pi value is also given. So see, pi comes in between the two pKa values, pKa2 and pKa3, and both of these are much higher than 7. So that means this is a basic amino acid, which is true because this is arginine. It is supposed to be basic, right? Now what are they asking you? Which of the following ionic structures would be expected at pH 8? So pH 8 means it is between pKa1 and pKa2. So what structure would be expected? Right. So think about it. Based on your understanding until now, you should be able to answer this. Initially at pH 0.5, only the positive charge would be there. Baki all the COO minus would be COOH, uncharged. At pKa at pH 2.1, after pH 2.1, okay, so we have got one answer, that is B. Okay, great. So after pH 2.1, what happens? You get one negative and one positive charge. No, sorry, not one positive. Arginine is having how many NH2 plus groups? See, this is the charge that you're having, right? So it is initially, so it is having two positive charges. So initially it is this structure. Then after pKa 2 it will become which one? This is what you are supposed to find out. Okay. Now. So the answer is D. So initially it was this structure. So it is having +2 charge at pH less than 0.5. After pH 0.5 and between pH 1, it is having this particular conformation, this particular state. And that is why the charge is actually, what is the charge? The charge is +1. So at pH 8, the overall charge of the amino acid would be +1. So pKa is 9. That means after pH 9, the thing is going to change. But from 2 to pH 9, it is going to be the same. So it would be +1. Okay, so once the pH crosses 2.1 and until it reaches 9, the ionic bond would be +1. Okay, I hope that is clear. Now we'll go to another question, which is Henderson-Hasselbalch equation. And what does this say? Oh, sorry. I think the answer is given. No. Okay, you try it. So here are the, now what is, you understand what Henderson-Hasselbalch equation is, right? So from this Henderson-Hasselbalch equation, what can you derive? So the first one is that it does not explain the behavior of di, tri basic acids or weak acids, right? It is usually useful with solution of acetic acid and hydrochloric. Now these are one weak acid and one strong acid. Relates the pH of a solution to the pKa and concentration of acid and conjugate base. This is correct. Allows the graphic determination of the molecular weight of a weak acid from its pH alone. See which of these are correct. So we look at the answer. The answer is 1 and 3. 1 we saw, 3 is actually the definition of this, and it does not explain the behavior of di or tribasic acids. For example, this particular curve, right, it only talks about one conjugate acid and its conjugate base, right? So it does not explain the behavior of this di and tribasic acids. Okay, so now we go to another question. Now I told you that histidine and its pi is very significant in nature. Look at this question and try to understand. So the pKa2, that is pKa for the side chain of histidine, is 6 when it is present in the isolated state. Considering a specific protein with histidine at its active site, the pKa is expected to be less than 6 if the active site has more positive charge, greater than 6 if the active site has more positive charge, less than 6 if the active site has more negative charge, same like isolated histidine, that is 6. Now which one do you think would happen? Okay, let's look at the answer. The answer is less than 6 if it has more positive charge. So when histidine residue is present in an active site having mostly positive charges, the pKa2 becomes lower than 6 because positive charges in the active site destabilize the positive charged histidine side chain. So a higher concentration of protons, that is lower pH, is required to protonate this histidine side chain, resulting in a lower pKa. So these are some of the things that you will know only when you practice these questions. Okay, so these are all previous year's questions. All of these questions were there in the previous year NPTEL course, Fundamentals of Protein Chemistry, and other courses also from where I have taken the questions. Some of these are new questions, but then mostly they are previous year's questions. So only when you practice these questions, you will come to know about some new things. Okay, this is one of them. Right. So now this is a very interesting thing. So see, this is a tripeptide. So there are 3 amino acids, one aspartate, one glycine and one lysine. So these 3 are joined together and all the pKa values are given, and they are asking you to find the pi of this tripeptide. So how do you do it? At different pH, you calculate the charge for the different charge for the tripeptide. Then what you do is you take the pKa which are on 2 different sides of the pH at which 0 charge is coming. So in this case it is 2.18 and the other one is 9.6. Sorry, this one is 9.6 and 3.65. Right, so 3.65 and 9.6. So you take these two and you take the average. This is exactly how you do it. Okay, right. Acha. So now we have discussed enough about amino acids. Now these amino acids actually join together by forming peptide bonds to form proteins. How are proteins formed? They have the primary structure, which is the sequence of the amino acids, and this is the signature of the protein, and then these structures hold together through different kind of interactions. There can be hydrogen bonding, which is majorly seen in case of the secondary structures. There can be different hydrophobic interactions. There can be different side chain interactions, and also disulfide linkages and many other interactions that give the natively active state, which is the tertiary structure. But in some proteins, for example hemoglobin, this tertiary structure is not active on its own. It needs to come together with other monomers and it forms a tetramer in case of hemoglobin. So it can form a dimer, it can form any structure, and that is called the quaternary structure, and it becomes functionally active. Okay, so now we will look at the peptide bond. So peptide bond is formed because of a condensation reaction. The carboxylic group and amino group come together, give out a water moiety and join together, and the peptide bond is formed. Now this peptide bond is very unique because normal CN bond length is much higher compared to that of the peptide bond. That is because the COO group is present. Because of the CO group, there is constant exchange, and that is why this partial double bond character is seen in the peptide bond. That is why peptide bond is relatively lesser in size compared to normal CN bond. Okay. Because of this shared bond electrons, what happens is the peptide bond becomes relatively rigid and prevents free rotation. This rigidity contributes to the overall three-dimensional structures of the proteins. Okay. So now you can imagine it this way, that this polypeptide chain is there. It is connected by different bonds. Okay. Now the planes actually keep on rotating this way. And this is how the different amino acid, let's take one amino acid as one plane and another amino acid as another plane, and this is how they keep on rotating. So if the rotation is between the N and C alpha bond, N and C alpha of the same amino acid, then it is called phi. If the angle of rotation is between the C alpha and C, so C alpha and C of the same amino acid, then this is called psi. And there is omega. Omega is actually rotation around the peptide bond. Now this is relatively less. So this is between the adjacent amino acids. Peptide bond connects to amino acids. So that is why it is between adjacent amino acids. Okay. So the planarity of this peptide bond is actually partial double bond character. So it is planar in nature. And this restricts the omega to 180 degree in very nearly all the main chain peptide bonds. In rare cases, omega is 0. That is for a cis peptide, which usually involves prolines. Now, what is cis peptide and what is trans peptide? For example, this is a carbon atom. This is the carboxylic group, like oxygen atom. Okay, CO is the carboxylic group. And then this is connected to the peptide bond. Let's say this is the peptide bond and this is the N. This is the N atom and this is the H atom. If it is there this way, one is pointing upwards, one is pointing downwards, this is called the trans peptide. But then if it is present this way, this is called the cis peptide. Both are in the same orientation. So cis peptides are normally seen when this is a proline and this is a, let's say, a different amino acid or proline. But then normally trans peptides are seen in nature. Okay. Now, based on the possible values of these phi and psi angles, peptides can be grouped, these amino acids in different polypeptides. They have been rigorously studied, and based on these values, they have been grouped into different secondary structures. Beta sheet, right-handed alpha helix, left-handed alpha helix, and it has been mapped and it has been seen that for a beta sheet, only this particular range of phi psi angle is allowed. Otherwise, it becomes a random coil, right? And for, let's say, right-handed helix, only this to this range is allowed. For left-handed helix, this particular range is allowed. So, all of the list is given here. Now, Ramachandran plot is something from where you will get questions every single year. So, this is something that you cannot ignore. You give me 1 second, I'll come back. Okay. So, Ramachandran plot is something that you have to understand properly and you will have to remember the values a little. Okay. Now, the Ramachandran plot is commonly used tool to visualize the distribution of phi and psi angles in a protein structure and assesses the allowed regions of conformational space for a given amino acid. Now, glycine is very highly flexible and that is why it can be anywhere on the Ramachandran plot. It does not have a defined location. But then the more rigid amino acid, for example, proline, these are having more restricted place in the Ramachandran plot. Okay. Right. So remember to learn about Ramachandran plot, I would recommend the books Voigt and Voigt's Biochemistry and Leninger's Principles of Biochemistry. So all the books and everything, all the materials that you should refer to are given in the reference slide at the end of the PPT. Okay. So the PPT will be available for you to download. So please go through all those books. Now try to answer this question. Which statement about the peptide bond is incorrect? It is stronger than an ordinary single bond. It has partial double bond character. It is weaker than an ordinary single bond. So see, 1 and 3 are contradicting one another. So which one do you think is incorrect? Excuse me. D. Okay. Acha. So C is the right answer, because which is incorrect, this is weaker than an ordinary single bond. That is incorrect. It is stronger and it has partial double bond character. Okay. Now we go to the next question. Which of the following which of the following combinations of torsion angle are correct? Now this is something that looks difficult, but it is simple. So see, for peptide bond, it is always omega. Okay. For NC alpha, it is phi. And for C alpha and C, it is psi. Alright. Just remember this much. And you also have to remember the values. But this is the basic that you can remember. So see, peptide bond is omega. NC alpha is psi. And sorry, this is phi. And C alpha C is psi. Okay, this is what you have to remember. Okay, now we will go to spectroscopy. We will just go through some small basics of spectroscopy, and then in the later classes we will properly read about spectroscopy. Okay, so absorption spectroscopy. Now that we are talking about amino acids and proteins, you know that we work with proteins in the lab. You have learnt about there are so many drugs, there are so many different cell studies, different, let's say, there are so many things, protein folding, so much studies are done with proteins, right? So of course people should be able to quantify proteins, otherwise how can you say how tightly it is binding, how much protein is required for a particular, let's say, drug as a drug candidate, etc. So definitely people are able to quantify proteins, and they're also able to quantify nucleic acids. But how? That is what we are going to study. There are many reactions, many different biophysical techniques that utilize the property of absorption and fluorescence in proteins, and that is how we get to find so many different biological answers to different biological problems, right? That is why it is very important to understand spectroscopy. So interestingly, different substances can absorb and emit light at characteristic wavelength. So this is the basic principle of spectroscopy. So different substances absorb and emit light at different wavelength. Okay, so spectroscopy is the technique to study the interaction between matter and electromagnetic radiation. It involves analyzing the spectra of the emitted, absorbed or scattered light to gain information about the composition, structure and properties of the materials. So light source gives out light of different wavelength. This passes through a monochromator. So monochromator is also like this window which allows light of only one particular wavelength to pass through. Let's say yellow light or blue light, some light. Okay, and then it reaches the sample, and the sample gives out, let's say, light of a different wavelength. Then that is detected by the detector, and we analyze that and get the information about the sample. So absorption spectroscopy is a biophysical technique used to measure the absorption of electromagnetic radiation by a substance as a function of wavelength. The basic principle of absorption spectroscopy, UV visible spectroscopy, involves passing light through a sample, measuring the amount of light absorbed at different wavelength, and the extent of the absorption is related to the concentration of the absorbing species and characteristic of the molecular electronic structure. So this works on the principle of Beer-Lambert's law. Absorption spectrometer is the machine, the instrument which uses this particular absorption spectroscopy. Right, so Beer-Lambert's law is something that is very simple, but then it gets asked in every exam almost. There will be a lot of questions, numerical especially, based on Beer-Lambert's law. But it is very simple to understand. We'll look at this in this PPT. Okay, now one interesting thing is that these amino acids in proteins, they can naturally act as chromophores. So what is chromophore? It is actually a part of a molecule responsible for absorbing light in the UV visible spectra. So UV visible means visible region in the spectra plus UV region, due to conjugate double bonds in the aromatic ring. Now if you remember the structure of tryptophan, tyrosine and phenylalanine, there was this single bond, double bond, single bond, double bond. This is called conjugate double bond structure, right, in the aromatic ring. Because of this, they are able to absorb light in the UV and visible region of the spectra. So in proteins, these aromatic amino acids are called chromophores. Tryptophan absorbs light at 280 nanometer, very strong absorbance. Tyrosine at 275 and phenylalanine at 260. These residues absorb UV light, allowing the protein quantification using A280 nanometer. So what is this A280 nanometer? We'll look at Beer-Lambert's law and then you can figure out what is A280. So A280 is actually absorbance at 280 nanometer wavelength. So 280 nanometer means you can understand that this is definitely UV light. It is not visible light, right? Now in DNA, nucleotide bases are aromatic and act as chromophores. So nucleotide bases, for example, adenine, guanine, these are also having these conjugate double bonds, right? So adenine, guanine, cytosine, thymine, uracil. In RNA, all of these absorb light at 260 nanometer, which is used for quantifying them at 260 nanometer, right? So now another interesting thing is so this is what a chromophore is. Chromophores are naturally present in proteins and DNA. Remember this. Now we talk about fluorophore. Now what is fluorophore? So fluorophore is a molecule that absorbs light at one wavelength and emits it at a longer wavelength, higher wavelength. So In protein, intrinsic fluorophores are tryptophan. This is absorbing light at 280 nanometer but releasing light at 350 nanometer. Right, tyrosine and phenylalanine. So intrinsic fluorophores or fluorescent dyes. Now there can be extrinsic fluorophores also. For example, any dye. For example, Cy5, Cy3. These are all dyes. FITC, Alexaflor. So you might have read about these in your cell biology like studies, where they use all of these different dyes to visualize proteins and all in the cell movement of proteins, right? So these can be covalently attached to proteins for imaging or FRET. Now in DNA bases are not fluorescent. DNA bases are not fluorescent, but then you have heard about ETBr, ethidium bromide, which is a dye which intercalates between the bases of DNA, and when you run it on an agarose gel, you are able to see this orange band, which is actually your DNA, right? So these are extrinsic fluorophores. This is very useful in different kinds of reactions. Okay, now we move on. So we'll go to Beer-Lambert's law. So in Beer-Lambert's law, see what is happening. This is the cuvette. Some of you might have done biochemistry practicals in your college. So there will be this glass container or plastic container, and they will tell you to be super cautious with cuvettes in college because cuvettes are very expensive. And in the cuvette, your sample will be kept, the protein sample, and you will place it into this machine. And then what happens? Light passes through it, and then the length of the cuvette is taken to be this small distance through which the light travels. This is taken to be 1 centimeter, and then it passes. So this Beer-Lambert's law, Beer and Lambert are two different people. There is a separate Beer's law and there is a Lambert's law. So Beer's law states that the concentration and absorbance are directly proportional to each other. Lambert's law says that that the absorbance and path length are directly proportional. Path length is this L, which is the length of the tiny diameter of the cuvette. Okay, so Beer-Lambert's law states that for a given material, the sample path length and concentration of the sample are directly proportional to the absorbance of light. Now, this is telling that this is directly proportional. Now, to make something that is proportional into something that is equal, you have to multiply with a constant. So, here the constant is actually molar extinction coefficient, which is represented as epsilon. This depends upon the number of tyrosine, tryptophan and phenylalanine present in the protein, right, into the individual molar extension coefficients of these particular amino acids. So that is how it is done. So A is equal to epsilon into C into L, that is the formula, and epsilon is equal to the molar extinction coefficient of tryptophan into number of tryptophan plus that of phenylalanine into number of phenylalanine plus that of tyrosine into number of tyrosine. These values will be given to you, and the numbers will also possibly be given to you, but you will be asked to find out a lot of absorbance and concentration values. So absorbance is equal to log 10 of I naught by I. I naught is the light that is entering into the sample and I is the light that is emerging. So here absorption is happening. So the light that enters is not equal to the light that leaves because a lot of light has been absorbed, right? So that is all this is about, right? So now see, this is the instrument. So UV visible spectroscopy. So that was absorbance spectroscopy. This is UV visible spectroscopy. This involves measurement of the absorption of ultraviolet light and visible light. Light source is often tungsten lamp or deuterium lamp for UV radiation and provides a beam of light that passes through the sample. Monochromator is there to select the specific wavelength, either 280 nanometer or 260 nanometer. It can be any wavelength that you want to select. And then there is the sample holder and the detector which measures it. Okay, right. Now there is something very interesting. So what happens is, normally the absorbance that we are supposed to see, sometimes we don't see it. Sometimes there is a shift. So what is this shift? There is hyperchromic shift. Hyperchromic shift happens when a molecule undergoes change in conformation, interacts with other molecules, or experiences a change in the environment that leads to an increase in the absorbance or fluorescence intensity. There can be hypochromic shift, that is decrease in the absorbance or fluorescence intensity. This happens when there are changes in the molecule's environment, such as decrease in the solvent polarity or alterations in the molecular structure. High hypsochromic shift. So what happens here is there is this are associated with higher energy transitions. For example, a shift in the absorption or emission spectra towards shorter wavelength might occur due to an increase in the electron density or change in the electronic structure of a molecule. It goes towards the left. Bathochromic shift is decrease in the energy level. This shift occurs due to factors such as increase in the solvent polarity, change in molecular structure, or interaction with other molecules. So bathochromic is red shift and it shifts towards the right. Now there is this MCQ. So you can read about this in details, and I have included only the part that is required for this MCQ into this PPT, but you can definitely always read about it. Okay, so try to answer this question. There is A and then there is B. So from A to B this transition is happening. Which two shifts are involved? Okay, it should be easy to answer. Just now you studied. So one difference that you can see is this is definitely higher, and another difference is that it is moving towards the left. So then which two shifts do you see? Chalo, try to answer. Right, so one is definitely hyperchromic. Okay, you're saying it's B. Let's look at it. Yeah, very good. That is B, right? So why is it B? Because one, it's definitely hyper, and second, it's going towards the left. That is what? One, which is hypsochromic shift, right? So B is the right answer. Next, we move on to two other questions. Try to answer these. Which among the following amino acid has the highest molar extinction coefficient value? This also we learned just now, right? Try to answer. A, correct, very good. So which among the following is correct about the absorption spectra for amino acids? Now this you have to remember. Okay, what happens because of what? So that is something that you have to remember. It will be confusing, but once you start solving the questions, it will become relatively manageable. Let's look at the answer. Right, so see, for tryptophan, the value is actually 5500. Okay, but for tyrosine and phenylalanine, it is very less. And the answer is, which is correct, so hypsochromic occurs when polarity increases because of indole ring. Yes, correct. Right. Now, due to increase in polarity, conjugation is reduced. Consequently, the energy gap is increased, which results in blue shift. So, this hypsochromic is called blue shift, and this side it is bathochromic is actually red shift, and the band position moves to shorter wavelengths. You can actually read about this in details, and you can find out what is happening, why it's happening. Right. So now we go to another question. So these are the questions that you should be prepared with. Okay, you should be prepared for these kind of questions. This is a NAT, numerical aptitude test, which is very common in GATE exams, and even in CSIR they can ask you numerical questions. So you should be prepared for this. A peptide containing 85 amino acid residues with molar extinction coefficient is given. Okay, having a molecular weight of 10.2 kD. If the peptide contains 4 tyrosine with absorbance value this, then calculate the number of tryptophan and concentration of the protein solution. Extinction coefficient for tyrosine and tryptophan are this and this respectively. So what law are you going to use here? Which formula? a very simple question. You're going to use the Beer-Lambert's law here, right? A is equal to epsilon into C into L. So C is the concentration of the protein solution that they have asked you to find. Now right, exactly, Beer-Lambert, correct. So epsilon total is equal to extinction coefficient of tryptophan into number of tryptophans plus extinction coefficient of tyrosine into number of tyrosine. So using this, you find out the number of tyrosines that are, you find out the number of tryptophans that are supposed to be present. Once you find that out, you will find out the concentration. So L will be 1. L will always be 1 centimeter, and the extinction coefficient is given, and absorbance, I think it is also given. Right. So then that is how you actually find out. So this is how you are supposed to do it. So see, epsilon value is this, and it is having 4 tyrosine. So then 4 into this plus x into this, that is equal to this value. So you get x as 6, and from the equation A is equal to epsilon into C into L, the absorbance is given as C. The absorbance value is this, and therefore absorbance is equal to epsilon into C into L is always 1, and so you get the value of C from here. Okay. Now that was all for this session. Thank you for your time. So these are the different materials that you can refer to. I have used all of these to make these PPTs. Be it the figures, be it the text, mostly was taken from all of these different resources. Okay. Now you can go to this very interesting virtual labs, practical, like, what should I say? It's like a virtual laboratory where titration of amino acids, that experiment is done. So there is the theory portion. Theory, it is given in very high details. So whatever you've learned until now, it is given in much more details there, and we ourselves have developed this. So this theory portion is given, and then there is this practical like thing that is done in the form of simulation. So they'll tell you to add different amino acid solutions into a solution and all, and that is how you can actually titrate amino acids, and you can find out the PI of different peptides, dipeptides, tripeptides, etc. Okay, so you can actually check that out. Baki, everything is there, and yeah, these slides are available for download. They will be there in the NPTEL. I think they will send you this page where all of these links will be there to a particular folder, and from there you can download all of these slides. So all of this is downloadable, plus, okay, yeah, plus you can also get this entire recording on YouTube. Okay, the link to that will also be provided to you by NPTEL. Thank you so much. We'll meet next week again with different lectures. Thank you for your time. So if you don't have any more questions, we can end the call. Okay, thank you so much.